So passive transport. Passive transport proteins, which you saw before, basically give a specific solute a door into the cell so it can follow its own concentration gradient. So this uses diffusion. This is why it doesn't require energy. All you're doing is giving the solutes a path to follow their concentration gradient. Some channels are open, others are gated so that the cell only opens them when they need it on the inside. And ions and molecules move to the side of the membrane where there are fewer of them. So the cell can use this both to get things in and to get things out. So here's a picture of this. This is a passive transporter. Its protein is specifically designed to move glucose into the cell. Here we've got more glucose outside of the cell than inside the cell, so the glucose will follow its concentration gradient and enter the cell through this passive transporter. And in this case, when a glucose molecule comes in, it closes the door behind itself to let it squirt into the cell, and this is what keeps the cell from getting too much glucose. And here it has released the glucose and it has gone back to its original shape. So it's ready to accept another glucose molecule. Now, active transport needs energy because you're moving a solute against its concentration gradient across the cell membrane. So this would be like if you have a lot of one substance in the cell but you need more and you're pumping it in. ATP is used to power this. And one way that your body's cells have kind of adapted to be crafty is they use transporters called cotransporters, where they move two substances at the same time, either two different things in or one in and one out when they're burning the ATP. So they can do two things at once. Here's an example of an active transporter for calcium. In this case, the cell wants to move the calcium out of the cell, but there's more outside than in. So you burn ATP in order to shoot the calcium out. A cotransporter is the sodium-potassium pump. This moves 3 potassium out for every 3 sodium out for every 2 potassium in. And this one just requires ATP to be burned to kick out the sodium, and then the reload part with the potassium doesn't require energy. Now, what if the cell has to get rid of a lot of stuff at once? Waiting for active or passive transport to occur would take a long time. So the cell can use endo and exocytosis to speed things up. So I call this taking the bus. If the cell is surrounded by something that it really wants, like a bunch of glucose in this picture, part of the cell membrane can actually pinch off and bring the substances into the cell. This can also happen when substances bind with the receptor proteins. It's kind of like the cell is fishing for what it wants. When what it wants binds with the receptor protein, it engulfs it with this net of cell membrane and pulls it into the cell. In white blood cells, this is called phagocytosis, where white blood cells will actually engulf microorganisms, debris, or other particles and digest them. So here's an electron micrograph of this happening. Here's the plasma membrane, and then there's these little dots here are lipoproteins that the cell wants, and the cell actually will close around the lipoproteins to, get it in. Here's a cartoon of phagocytosis. Here's a white blood cell eating a big pathogen, that big brown thing. So the cell actually moves around the thing that it wants to digest, so it does the endocytosis. And then lysosomes, these white things here, fuse with the vesicle that the pathogen's in and digest it. So the cell then just either uses the digested material or expels it with exocytosis. And here's a cartoon of exocytosis. This is where a vesicle has formed inside the cell and it binds with the cell membrane and expels its contents. This is how, when the membrane proteins are made in the rough ER and the Golgi, They're actually put into a vesicle, which then binds on the surface and the protein gets stuck on. So here's a cartoon of that happening. These little white things are getting made in the Golgi. They float up. When they bind to the surface, they are now part of the surface. We talked about diffusion. Osmosis is the same thing as diffusion, but osmosis specifically refers to water. You could say osmosis is the diffusion of water. Water's concentration depends on how much other stuff is dissolved in it. Highly concentrated water is something like distilled water, where it's just water. Less concentrated water are things like seawater and the water inside your cells. Now, when cellular transport goes bad, bad things happen. And one of those bad things is the genetic disease cystic fibrosis. CFTR is a transport protein in the membrane of cells that lines the passageway of the respiratory system, as well as other epithelial or skin-like tissues. This protein transports chlorine items out of the cell. So this is what it looks like. It puts the chlorine out of the cell. Osmosis then drives water out of the cell. So it's used to make mucus. This is the mutated form of the protein. Individuals with cystic fibrosis have a mutation that causes part of this protein to be missing. Now the cell can't expel water as well as it should. This means that the mucus of CF patients is really sticky. And this mucus causes their respiratory tracts, causing them to have constant infections. Most patients with cystic fibrosis end up dying in their 30s, although medicine is working to extend this. Now, if we look at a system like this, where we have a semi-permeable membrane and we have solution on either side, the water is blue, the solute or the salts dissolved in it are red, and in this system, the water can pass through the membrane, but the big red salts cannot. If a substance is isotonic, both of the solutions on either side of the membrane have the same solute concentration. A hypotonic solution has a lower concentration of solute, so this side would be hypotonic. A hypertonic solution has a higher concentration of solute, so this is a hypertonic solution. Water will diffuse or osmose from a hypotonic to a hypertonic solution. So the water goes from the hypotonic solution to the hypertonic solution, and this is what you get in the end. These are two isotonic solutions, and you can see that the ratio of water to salt is about the same. The water moved down in this tube and up in this one. This is very important, and you may see it on the exam. So here's another example of this. Let's say you have a bag of dialysis tubing, like you'll have in Lab 3, that contains a 2% solution of sucrose. If you put it into a beaker that contains a 2% solution of sucrose, the two substances inside and outside the beaker are isotonic. So this means that the concentrations are the same. There's no concentration gradient, so water won't enter the bag or leave the bag. If you put a 2% sucrose solution into a hypertonic 10% sucrose solution, there's more sucrose outside of the bag than there is inside. So there's a higher concentration of water in the bag than in the outside of the bag. So the water is going to leave the bag, leaving the sucrose behind. Then that makes our bag shrivel. In this last example, if you put the 2% solution of sucrose into water, there's no sucrose outside of the bag and there's some inside. So the water is going to go into the bag, trying to even out the concentration gradient. Remember, in this example, the sucrose could not travel through the membrane. This also happens in your body. Red blood cells are supposed to look like this. They're in an isotonic solution and they do not change volume. If you drink salt water, the red blood cells in a hypertonic solution, so salty blood plasma, shrivel because the water diffuses right out of them. Also, if you put your cells in a hypotonic solution, the water will diffuse into them. So this is if there's too much water in your body, the red blood cells will actually swell and pop. Now, plants take up water from the soil via osmosis. So the plant cells are hypertonic compared to the soil water. This is what allows the osmosis to take place. The plants take up the water and swell until their cytoplasm starts pushing up against their cell walls. This is called turgor pressure, and it's what keeps the tomato plant up and looking good. When the plants reach their limit, they can't take up any more water. When the soil dries out, the concentration of salt in the soil water increases. The soil water becomes hypertonic, so it has more salt in it than the fluid in the plant cells. And osmosis actually happens in reverse, so the soil draws more water out of the plant. So this is why when you don't water your plant for a while, the turgor pressure will drop and your plant will wilt. This is what it looks like on the inside. Here are some plant cells. They're nice and happy. The cytoplasm is right up against the cell wall, so you have high turgor pressure. In this example, the cytoplasm is shriveled. It's not pushing on the cell walls. And now this plant is, you see these big spaces here. This plant is going to wilt. And that's it for today. Have a lovely day.